So we are going to do today anaplerotic reactions. What are they? Why are they required? And what is their significance? The anaplerotic reaction. To understand this, we must know that PCA cycle has got dual role. It is not only the central catabolic pathway, but it has got certain anabolic role also. The TCA cycle provides an important pool of intermediates: citrate, alpha-ketoglutarate, succinyl coenzyme A, fumarate malate, oxaloacetate. So these are important pool of metabolic intermediates which serve as precursor for various important compounds. For example, alpha-ketoglutarate. It is starting point for biosynthesis of a number of neurotransmitter. For example, excitatory transmitter. Neurotransmitter is glutamate. Inhibitory neurotransmitter is GABA, gamma aminobutyric acid. Likewise, succinyl coenzyme A is the starting point for synthesis of heme, which is prosthetic group of a number of important proteins like hemoglobin, myoglobin, cytochromes, uh, catalase, tryptophan pyrrolase, etc. And then some intermediates of TCA cycle can form certain non-essential amino acids. For example, oxaloacetate forms aspartate. Alpha-ketoglutarate forms not only glutamate, but also glutamine. And then citrate, it can very easily, with the help of a carrier, it can move out of the inner mitochondrial membrane, cleaved into acetyl coenzyme A, which forms starting point for synthesis of fatty acids and cholesterol. And of course, oxaloacetate, not only for aspartate, it can form via gluconeogenesis, uh, pyruvate, and then, various glucose, etc. So oxaloacetate is the starting point for gluconeogenesis also. So we have seen till now that TCA cycle, it has got an important anabolic role, or rather roles. So therefore, it is also called amphibolic pathway. Now here arises an important problem. Problem is that the TCA intermediates are flying off for biosynthesis of various biomolecules. So what can be the result? The result of that might be that the TCA intermediates may soon get depleted. What do we mean by that? For example, if oxaloacetate is going for gluconeogenesis or for synthesis of a non-essential amino acid, then gradually the concentration of oxaloacetate will fall. Likewise, alpha ketoglutarate agar kam ho gaya, ultimately it will lead to decreased intramitochondrial level of oxaloacetate. And oxaloacetate is the starting point for TCA cycle, the first intermediate. तो oxaloacetate की concentration अगर कम हो गई, then the first step of TCA cycle will be hampered and altogether stopped at a later stage. So the TCA cycle may come to a grinding halt. It may stop altogether. I repeat, concentration of oxaloacetate is an important factor for, is an important regulatory signal for TCA cycle. Oxaloacetate come ho gaya, TCA cycle also will halt ultimately, or it may be impaired initially. So therefore, it is necessary that the TCA intermediate should be regularly, they should be replenished. And how that is done? For example, pyruvate, we had written here, pyruvate can be converted to oxaloacetate with the help of an enzyme called pyruvate carboxylase. Oxaloacetate by transamination reaction can be generated from aspartate. Alpha ketoglutarate, again by transamination reaction, can be generated from glutamate. And likewise, succinyl coenzyme A can be generated from an amino acid called methionine. Fumarate can be generated from phenylalanine degradation. So we had seen that because of biosynthetic role of TCA cycle, various TCA intermediates are at risk of getting depleted, which may ultimately lead to sluggishness or halting of TCA cycle, a dangerous situation indeed. So therefore, certain filling up reactions, as shown by these blue arrows, are necessary, and these filling up reactions are called anaplerotic reaction. So to summarize, anaplerotic reactions, they are filling up reaction. And we have seen why they are necessary, because the TCA intermediates may fly off, as shown by these arrows, for biosynthesis of various compounds. So their concentration may gradually fall down. So to overcome any dangerous consequences because of depletion of TCA intermediates, these filling up reactions by, as shown by these blue arrows, are necessary. So I repeat the examples: pyruvate carboxylase generating oxaloacetate from pyruvate, transamination generating alpha ketoglutarate from glutamate, and oxaloacetate from aspartate, fumarate from phenylalanine, succinyl coenzyme A from methionine. So lot many others also are there. You can just see more examples yourself. So with that, we stop. Thank you so much.